Hello, I'm Paul Macklin. I'm happy to welcome you to the 2022 edition of the Virtual PhysiCell Workshop and Hackathon. This virtual course is a series of sessions with a number of goals. In the workshop, you're going to learn about agent-based modeling and biology in general. In particular, you're going to learn about how cells interact in multicellular systems biology and how we can write models to do this. We're going to learn to build and explore agent-based models using the PhysiCell system. That's kind of the main purpose of this course. And if you're a member of the hackathon, then we are also going to have you form a new team and build either a PhysiCell model of a particular multicellular system or build a PhysiCell tool that will help the community. Other goals include meeting other members of this community of cancer and multicellular systems biology modeling, and to identify new leaders in the PhysiCell community to kind of help grow this community of users and scientists as we go. In this particular session, session 0, because programming starts counting at 0, we're going to learn the motivation for and concepts of agent-based modeling. We're going to briefly survey some of the other approaches to agent-based modeling. We're going to learn about PhysiCell and its approach to agent-based modeling, and we'll show you a few examples and some other resources that will help you go through this virtual course. So one thing that really interests us as biologists, as scientists, as mathematicians and modelers is to understand how we start with individual cells that have kind of a limited palette of behaviors. They can grow, they can divide, they can change type or differentiate, they can die, they consume substrates in their environment, can secrete them as well, and of course metabolize them. They can signal to one another, they can mutate, they can even eat each other and do other things. And so here we get from single cell behaviors now to their interactions, that they can stick to each other and push each other around mechanically. They can predate each other, so they can actually eat one another, or they can fuse together. And they can send signals not just through secreted chemicals, kind of in a passive way, but very actively through contact interactions. And then you put these cells that are individually acting and then communicating into an environment with physical constraints, things like diffusion limits and diffusion barriers and mechanical barriers. And somehow, the combination of this relatively discrete set of individual behaviors and interactions combines to form the complex ecosystems of cells that we see in biology and disease. And so multicellular systems biology seeks to understand how the cell interactions and cell behaviors, as you put more and more cells together, give rise to these complex systems. And of course, as physicians and engineers, we want to not just understand the systems, but take that understanding and manipulate it to control the behavior of these multicellular systems, to build a tissue, to take an existing tissue and return it from a disease state to a healthy state. And so we are using these multicellular systems to understand how changing one or more parts of the systems, changing communication, changing behavior, can give rise and control the behavior of the system. And one analogy I like to use for multicellular systems in general is that the microenvironment, that the tissue and the chemicals in it is the stage, and the cells are the actors, and they're creating together, as they work together, a play upon that stage as they follow their scripts. But these scripts are based upon the stage. That is, what the cells decide to do with these actors is dependent upon the scene. And so if you change the stage, you change the behavior. And that the cells are not delivering monologues here. They talk to each other. And so the cell-cell communication interaction is really critical to what happens in biology. And then what can be very interesting is that the actors are not just actively, passively sitting on their stage, but they are interacting with their stage. They can rip up the stage and remodel it. And sometimes in disease, they can kind of go off script and make their own script. And that might be the examples that we see in mutations and evolution. And so, generally speaking, multicellular systems biology, it's our job as scientists to figure out what is the script by watching the play over and over and over again. You take the actors, you place them on a stage, you watch the play, you jot down what they do, and then you modulate a little bit. You change the scene and see if the behaviors change, and see if we can kind of tease out the script. And then if we can figure out how that script varies with the scene enough, with the chemical signals, with the contacts, with the mixing of cell types, then maybe we can rewrite the script and engineer the behavior of the system. And so agent-based modeling is a paradigm that helps us to understand and explain these complex multicellular systems. And in this idea, we're really taking it down to the basic level of the cell, to say, what are these cells? Can we model them as individual agents with their individual behaviors and then their interactions, and place them in a virtual tissue environment that models this stage with the players with their scripts. And so before we get into agent-based modeling, I'd like to just kind of point out some of the differences between continuum and discrete modeling. So discrete here refers to discrete mathematics. And so in contrast, a lot of you are probably already familiar with continuum models, that you have a model with a continuous variable, such as a cell population density, and it varies continuously, typically has derivatives. And so a classic example is exponential growth, that the change in the cell density over time is proportional to the cell population. Well, discrete models, rather than referring to the overall population, refers to the distinct individuals in the population which may be performing discrete events rather than continuous events. And so these variables tend to have discrete values, like Boolean values as a true or false, or integer values like 0, 1, 2, 3, or 4. You can't have 17.3 cells in the discrete model. You have 17 or you have 18. As an example, take that exponential growth, but now turn it into something discrete. You have a population x of t, which has integer values of individuals in the cell population, and for every one of those individuals, they have a Poisson process with a rate r that governs division. So between now, t, and another short time in the future, t plus delta t, each cell then has a probability, r delta t, of having a birth event that increases your counter, x, by 1. So there's a discrete version of that same continuum model. Agent-based models are a form of discrete model where you simulate the individual cells and their behaviors. And so it's a little bit more complex than just a counter. You're modeling a broader palette of their behaviors. These have lots of names. Sometimes they're called individual-based models or cell-based models. And so you'll have often an agent-based model that models like division, birth, death, and movement. And then on the top of that, you'll add a continuum model for the environment, things like a diffusion equation for oxygen or a growth factor or a drug. And so you'll often call this a hybrid discrete continuum model, where you have the discrete cells with a continuum environment. And so kind of more from a computer science perspective, these agent-based models, where they have their own individual behaviors and states, are really ideal for something called object-oriented programming. And so you focus on the individual cells. You're going to come up with kind of like a general class for the cells. Like, here's a template for all of my fibroblasts. Here's a template for all my cancer cells. Here's a template for all of my E. coli. And then the individual cells are an instance of this class. And so that agent class will have its own data and its own behavioral rules. And then you have a single copy for each of your individual cells. And so agent-based modeling allows us to move just a little bit closer to the biology by directly linking what we see in an experiment or in a textbook or in a paper, and directly carrying that over to our software object, that you can model the individual cells and their behaviors as software methods, and then you can apply them to particular rules, to particular problems, by choosing the hypotheses and turning them into the rules. So you might have a hypothesis that says maybe oxygen limits cancer growth. And so when you have low oxygen, proliferation reduces, high oxygen, maybe it increases, and then maybe low oxygen might trigger changes in behavior. Well, those can all become hypotheses that become rules in your model. You can tailor those rules down to the level of detail that you have. So if you have a whole bunch of genomics information or metabolomic information, maybe you might choose to embellish or increase the scope of your model by adding a molecular scale model, like say system differential equations that model the data that you expect to get and those observations and connect it to the cell behaviors. So in a typical agent-based model, they really have a fairly typical flow of the program. You're going to start with their setup. You're going to read in, say, some kind of a parameter file that gives you some notion of everything that's going to be in your model. Say the birth and death rates of your cells, maybe how many different cell types you have and how many of them, maybe where they're placed, some information on your environment, like say oxygen and glucose might have specific diffusion and decay rates and boundary conditions and initial conditions. And so you're going to read those parameters, start setting up your environment, like setting up those diffusion equations. Then you're going to set up your cell agents. Say, what kind of cell types do I have in my model right now? Maybe I have fibroblasts and cancer cells and macrophages. And so you might define those cell types and their default parameter values. And then when you're ready, you have the environment ready, you kind of have your cell types ready, and then you're going to start placing them in your virtual environment to initialize your model. And then you're ready to go into kind of your main program loop. For every single time, we're going to go and update the environment. So freeze the cell positions, have them do their secretion uptake, whatever they need to do, solve your reaction diffusion equations, solve your tissue mechanics as you need to, then freeze the environment and then go and update your cells. Sample their environment. Based upon what they see in the environment, update their parameter values that help them advance through their processes like birth, death, mechanics, and motility, and then run all those processes. And that will update the cell positions and states. And then freeze that, update time a little bit, say I advance by delta t, and then go back and keep repeating. So you do update the environment, update the cells, keep going until you're done with your simulation. There are a lot of kinds of agent-based modeling approaches, and we won't go into them in great detail here, but notice that one of the key distinctions is whether this model is lattice-based or off-lattice-based. In a lattice-based model, which might be familiar like, say, Conway's Game of Life or cellular automaton model, cells have discrete positions that can be at different grid points and move left, right, up, or down, diagonally, for example, but they'll have rules that kind of model these discrete jumps in positions, and that's a lattice-based approach. And there are a lot of successful techniques to use them. Off-lattice models include PhysiCell, where you don't have discrete positions. Instead, you say, here's my cell center, and I'm going to update that cell center's position based upon a balance of forces acting upon the cell. So a little bit more physics-driven. And there are a whole lot of ways to kind of get into varying levels of detail for these two kind of classical approaches of lattice or off-lattice. So if you'd like to see a deeper review on that, and I encourage you to look, we did write a review of cell-based models in the cancer modeling context, and you can see that at JCL Clinical Cancer Informatics, this little hyperlink over here, which will be included in the slides. So where does PhysiCell fit into this? PhysiCell is an off-lattice center-based model. The resolution of the model is one agent per cell, although there are some tricks where you can use bigger agents that might model collections of cells if you need some efficiency, or you might break an agent up into smaller agents and find a way to kind of connect them together to model cell shape. That's not as typically needed, but it can be done. PhysiCell then will couple these agents with partial differential equation models of the environment to make it a hybrid discrete continuum model. So we will model reaction diffusion of your chemical factors in the environment and connect them to the cell agents. And then we can add ODEs and other technical techniques like Boolean networks to model the dynamics and details of individual cells. And so you can really make this truly multi-scale, where every cell agent might have an embedded, smaller scale, intracellular model that drives the cell behaviors based upon what they see in their environment. And so to kind of get a little bit more in the depth of our toolkit, the first thing we built, realizing that biology requires, maybe diffusion of oxygen, glucose, some metabolites, maybe a handful of signaling factors, and one or two drugs that might introduce in the environment, you're easily looking at 5 to 10 diffusion equations in a typical problem. And so we had to do diffusion in 2D and 3D. So we developed something called BioFVM, which is a finite volume reaction diffusion solver that works on desktop workstations or on a cluster, and uses a variety of techniques to get efficient solution of a whole vector of reaction diffusion equations simultaneously. And in particular, we developed that so that you would have kind of like these proto-agents of cells as acting as point sources and sinks in the environment. So you have the reaction and diffusion of these factors with boundary conditions and initial conditions you might expect, and then cell-based point sources and sinks so they can secrete something to the environment or uptake it. And that's all open source. It works across platforms on Windows, on Linux, on Mac OS, where you can do your development on your desktop, and then when you're ready, take it on over to a cluster to run high throughput studies. And we published that several years ago now in bioinformatics. And PhysiCell then takes that diffusion equation, the stage, and adds the biology back into those point sources and sinks in the diffusion solver. That now they have off-lattice positions, but mechanics-based movement. Every cell has its own individual behaviors like cycling, death, motility, mechanics, searching for interactions. And the neat thing about this is it's very extensible. You can add custom variables or custom rules or data on a cell-by-cell basis and even change them as you go in your simulation. And so this code is also cross-platform. You can run it on Windows, you can run it on Linux, you can run it on Mac OS. And then when you're done, building your model, you can go send it off to a cluster to do high throughput studies. And this was published in PLOS Computational Biology several years ago. And we were very fortunate to be nominated for and receive the Computational Biology Research Prize for Public Impact for this work. So a typical model here is this one right here. Kind of shows you how very basic hypotheses can come together to give you some neat emergent dynamics. So this is a 3D cutaway view, and these cells are colored from blue to yellow. So each one of these dots is a cell, and the little dots inside are the cell nuclei. So we actually can model the nucleus and cytoplasm separately if you want to. The cells are colored blue to yellow based upon how aggressive they are. So yellow cells proliferate faster than blue cells. And of course, properties are heritable. So if a cell divides, it passes on its properties to its daughter cells. So that's kind of a key feature in an agent-based model. You can see now, because every cell agent can have individual properties, we can model the heterogeneity in this process. So the basic hypotheses in this model is you have oxygen kind of as a driving growth substrate, applied as Dirichlet conditions on the outer edges of the 3D domain. Oxygen diffuses into the environment, cells consume oxygen and deplete it, and so you get the emergence of an oxygen gradient as they get deeper and deeper into this tumor. And the more oxygen the cells have, the faster they can proliferate, and the less oxygen they have, the less they proliferate. And if oxygen gets too low, they become necrotic, so they start having a death based upon energy collapse. And you'll see those in a moment as the brown cells. So look here and see how yellow cells give birth to yellow, blue cells give birth to blue. I'm going to let this advance just a little bit. Now you can see in the center, these brown cells emerge. So now you have oxygen depleted in the center and the formation of a necrotic core automatically from these just very basic assumptions. And of course, the other background processes like mechanics going on. The other interesting thing, of course, is because yellow cells give birth to yellow cells and they're proliferating faster, you see these foci of faster proliferating cells emerging as subclones within this tumor. The other interesting thing to watch is the nuclei to kind of get a sense of the mechanical fluxes in this problem. On the outer edge, as you might expect, when a cell divides, it's easy to push the two daughter cells into this fluid-filled, unoccupied domain. But now suppose you have a cell division closer to the middle. Those two daughter cells can either push against all of these cells to get out, or it can be pushed towards the center. Well, it's mechanically favorable to actually have a big flux of cells moving to the center of the tumor, particularly as this necrotic core emerges. So let's watch and kind of see the movement of those cells. See how these guys are kind of being pushed into the necrotic core by the mechanical fluxes. Now, another interesting emergent feature. The necrotic core is growing because more and more cells are dying. Kind of have these foci emerging, and then the blue cells kind of squeezed in between them because they're proliferating less quickly. The other thing you see here is the necrotic core is kind of opening up these shadowy chasms, and that's because it's mechanically unstable. You have cells that are still adherent, but they're shrinking because they're losing volume as they degrade. And so you get this network of chasms forming in the necrotic core. And that's actually something we see in organoid models, particularly with Shaman Mummenthaler and her work at the University of Southern California. So there's just an example of a 3D tumor with very basic assumptions, and you kind of see then the yellow cells are competing and taking over. So in a PhysiCell model, you have a combination of factors in a typical physical model. You have your microenvironment, which is your stage, where we have a number of diffusible substrates, each which will have its own diffusion coefficient, decay rate, its boundary conditions, which can either be held fixed like a Dirichlet condition or zero flux like a Neumann condition. And these are completely defined in a configuration file. And then on top of that, we're going to start defining one or more cell definitions. These are like the types of players, your fibroblasts, your cancer cells, your macrophages, your E. coli, your CD8 positive T cells, where each cell definition will have a name, which will help you search for it, and then a default phenotype, which is kind of like the set of default parameters for its behaviors. Those can also now be defined in the configuration file. And then when you start the model, you'll have individual cell agents that are individual copies of those templates or those cell definitions. And say, for every cell agent, what type am I? What are my state variables, like my position, mechanical pressure, and who I'm interacting with? And then you have your phenotype, which kind of sums up and bundles the parameters, as well as any custom variables, and then functions that are attached to the cell that act upon the phenotype to change the behavior based upon your environment. So the entire workflow really is centered around the cell's behavioral phenotype. And we bundled the parameters for cell cycle progression, the parameters for death, parameters for volume, for mechanics like adhesion and repulsion, motility, secretion and uptake, and other cell-cell interactions, which would be really important in, say, immune type models. And so we kind of bundle these up hierarchically so we can very easily copy the whole set of the cell's behavioral parameters from one cell to another, or to change them very easily. So you can modulate these always-on processes of volumetric growth, of motility, of mechanics, and on a moment-by-moment basis, you can stack the environment and use your biological hypotheses to drive rules that change the parameters of how the cells are behaving. A new feature we introduced just this year, which I think is going to really change the game on how we model, is something where we have a notion of signals in the environment. These are basically any input to a subcell model or rule that says things in the environment that can influence your cell behavioral phenotype. And then we have behaviors. These are the parameters in your phenotype, like, say, cycle entry, or your apoptotic death rate, or your mechanical adhesion strength. Those are the behaviors that can be modulated. So those are like the outputs of the cell rules. And then we have dictionaries of behaviors and signals. They're automatically generated by PhysiCell at the beginning of your simulation. You can easily query the name of a signal by its plain English name. So you can actually say, get my value of oxygen with a function, and it will dump it out, and you can easily access it. So the syntax becomes very, very simple and intuitive. Then you say, okay, I want to get my default behavior from my cell line, my cell definition for, say, cycle entry. And then I want to modulate that parameter based upon my current signals and write my actual cycle entry rate, which might be increased or decreased. And then I'll write that value back to my cell using my dictionaries again. So this will really make it much easier and faster to build models. One other thing to note is that there are a variety of timescales to help make PhysiCell just a little bit faster, realizing that biology is very multi-scale in space and time. And so diffusion is happening very fast all the time, so it has the smallest time step size, by default 0.01 minutes. Mechanics is like the next fastest process. This kind of cells are moving around on a minute by minute basis, so the default time scale is 0.1 minutes to resolve that time scale. And then things like volumetric growth, cycle entry, that tends to have hours-long time scales. So we use a 6-minute default time step. And so by not doing everything at every time step, but kind of dividing up the work, allows us to take advantage of this multi-scale aspect of the cells in time and make this computations just a little bit faster. I'd like to point out that the intracellular models can have their own step sizes. And so we'll talk more about that later this week. So now I'd like to pivot and just show a few recent examples of agent-based models you've built in PhysiCell. So first example is drawn by some recently published work by Yafei Wang, who just graduated from our lab this year, looking at the interaction between colorectal cancer cell micrometastases and the surrounding parenchyma or liver tissue that they're metastasizing into. And you can get this paper open access at Scientific Reports. So one question that we were very curious about when we were working with collaborators was that you have a metastasis arriving in a tissue, and it has to contend with the tissue that's already there for it to successfully colonize and grow out into that tissue, and basically it has to replace it. And so we asked kind of the basic question saying, let's not worry about the fancy molecular biology for now. Let's just worry about the contact interactions. Say, if a tumor cell is growing into, a bunch of liver tissue and it is imposing mechanical force on the liver tissue, how does it affect the liver tissue? So that liver tissue should be displaced. On a short time scale, it ought to be an elastic deformation. So it kind of gets displaced, but it wants to go back to its resting configuration. But if you leave that liver tissue, that little tiny bit of liver tissue deformed for long enough or displaced for long enough, eventually you'll get a mechanical relaxation or a plastic reorganization. So on short times, you push a little bit of liver tissue out of the way to make room for the metastasis, and it pushes back elastically and puts a force back onto the tumor. But if you keep that displaced for long enough, that can kind of impose some biological stresses on that bit of liver tissue, and maybe it can't withstand it, so it dies and gets out of the way, or plastically reorganizes to reduce the stress. And then, so there's one way coupling, that the tumor's imparting forces on the mechanical forces on the tissue, displacing it, and imposing some stresses on it, biological stresses on it, and it will either succumb to that and die, or plastically reorganize and in order to deal with it. And on the flip side, then that surrounding tissue is imposing compressive forces back onto the tumor. And we know that tumor cells can downregulate cycle entry based upon mechanical pressure. And so that's the flip side of it. So we have that the birth of tumor cells, the metastatic niche, is driven by the availability of oxygen. So the more oxygen you have, the greater the cycle entry rate, but it's downregulated by pressure. So if your pressure goes up, your cycle rate goes down. And if it's high enough, you can just get an arrest of cell cycling. So when we model this, so here is a tumor metastasis growing into a large chunk of liver tissue. And the cells that are yellow are the cells that are experiencing high pressure and arresting their cycle. So you can see at the beginning, right, there's a lot of pressure kind of building up in the center of the tumor until it gets big enough to get necrosis or death here in the center of the tumor, which temporarily releases that pressure. So you can see here that on the inner edge here, we kind of have blue, where it's not high pressure, but it's also not high oxygen. So it's lower proliferation here, kind of an annulus of high pressure because you have proliferation and all this stuff. And then on the very outer rim, kind of a blue region of lower pressure. And as the tumor grows, you kind of see this type of pattern stabilize. If you were to take a cross-section of the tumor, look at the tumor-liver interface, see how the pressure kind of is a little bit lower at the leading edge. That's because you have some death in the liver that kind of locally relieves that pressure. Then it builds up as you go backward in the tumor. And then the pressure goes back down as you get towards that necrotic core. Then on the flip side, if you measure kind of the mechanical stresses in the liver tissue, you see how it kind of propagates and dissipates into the tissue. So as you might expect, the pressure and displacement is highest, or the stress is highest, in the liver tissue just immediately surrounding the tumor. So that's where you're most likely to have the liver cell death. and it dissipates surprisingly far into the tissue. So, the interesting thing is if you have just the right balance of elasticity of the liver tissue so that it can kind of put forces back onto the tumor, if the plastic relaxation is slow so that pressure is sustained, and if the liver cells can tolerate that displacement, so they really keep up basically the counterattack, you can get large regions of your invading tumor that undergo high pressure and pressure rest. So you can get, for certain values of parameters, a lot of compression in the surrounding tissue that compresses the tumor and keeps it from growing large. So you have these micrometastases that just reach kind of a mechanically dormant state. And if you run the tumors long time, they will actually withstand that. So we actually ran an experiment. So control, simulate 90 days, get the small metastasis, simulate another 90 days and see that it really stays in that small dormant state for a very, very long time. But now if you do a mechanical perturbation, like say change the elastic constant or change the tolerance for deformation, you have that previously dormant tumor just completely explode and start growing into the tumors. It's like a reawakening. And we know that we can get mechanical perturbations, like say from an injury or just over the course of aging. So you might have a lot of dormant micrometastases that this soil basically was unfavorable, but something changed and now conditions are favorable and that previously dormant tumor can start growing again, purely based on mechanical and contact effects, and not even starting to add the complex fancy molecular biology that we would further accentuate these aspects. So as another example, I'd like to highlight a project we did with Daniel Gilks's lab at Johns Hopkins University. And this is recently published in iScience, led by my postdoc, Aber Rocha, and with Anesh Goda at Johns Hopkins. And so Danielle and her lab has this beautiful mouse model where if you take a cross-section of a breast cancer tumor and kind of look from the outside in, so here's the outer edge of the tumor, and you can go in and you kind of see different layers of the tumor. And so they built this beautiful system where cancer cells will fluoresce red if they're normoxic, so they have normal oxygen, and then they will change and permanently change color to fluoresce green if they've been exposed to low oxygen conditions. So it kind of looks through the layers here. You have the greatest oxygen at the outside of the tumor, kind of transitioning to low oxygen, so it's green. And then this purple stuff is cells that haven't managed to do that switch yet. They are experiencing hypoxic conditions, but it takes time to go through the regulatory machinery to change from red to green fluorescence. And then you have this dead core at the center. And so one thing that we asked together as a team is that we know that cancer cells, when they're in low oxygen, when they're hypoxic, change their behaviors. In particular, they can increase their motility and try to escape the bad conditions. And so the question we had then is, what about those cells when they leave the regions of hypoxia, when they are no longer undergoing that stress? Do they retain the old hypoxic behavior of increased motility and change, possibly altered proliferation? Or do they instantly change back to their original normoxic program? Or is it something in between where they kind of have a persistent hypoxic phenotype for a little while until they switch back, kind of like a signaling lag, for example? And so Danielle's system was fantastic, right? You could actually track these questions because when you put these cells into hypoxic conditions, they basically perform their own gene editing on themselves, snip out the red gene and start permanently expressing their green genes. And that means that if you see a green cell, you know that at some point it's been hypoxic. And this allows you to track the long-term fate of hypoxic cancer cells. And so in our model, we start with kind of our classical assumptions that we have oxygen proliferation based upon the pressure and the oxygen availability, just like we showed you in that earlier model. And that liver cells, the live cells, then if they're normoxic, they would start off as red. But if they were exposed to green to hypoxic conditions, they would permanently change color to green using just a really basic ODE model of gene switching. And then the green cells would have an increased chemotaxis of oxygen gradients to kind of escape the low oxygen zones. And so then we added one more thing. We added something called phenotypic persistence, saying that if a green cell is left a hypoxic zone, what's the mean time it would take to reduce its motility? And so we would call that the mean persistence time, or the phenotypic persistence. So the interesting thing is, if you take those hypoxic cells, and if they instantaneously change their behavior back the moment they flee the bad zone, you'll never get invasion. And this kind of makes a lot of sense, right? That if you have, the green cells are migrating just long enough to reach the bad zone, there's a whole bunch of red cancer cells in front of them that still have plenty of oxygen. So you get this persistent ring structure where the green never leaves to escape the tumor. But of course, we know this is not correct. which means that the simplest hypothesis that cancer cells do not retain that a long-term post-hypoxic phenotype is false. It just can't be, because we know we see invasion. So now if you add on any amount of phenotypic persistence, you get a very different structure. You get these invading plumes of green cells that don't stop migrating when they reach the red material where there's plenty of oxygen. They keep going on through and forming these kind of structures that eventually can break out of the tumor and go and invade the surrounding tissue. And so as long as you have a non-zero phenotypic persistence time, accounting for that lag of behavioral, behavioral changes, you can get things that match what you see in experiments. And in fact, let me pause this for a second. So here's like a computational cross-section where we have this green plume kind of invading into the red tissue, and we see these experimentally in Daniel's mouse model. So we were really excited that some really basic assumptions in the agent-based model were able to then lead to emergent behaviors in this collection of cells that can be validated experimentally and give real evidence. And this really kind of gives the power of combining computational modeling with a novel engineered system. That the novel fate tracking of Daniel's system allowed us to see these structures for the very first time. And the computational model gave us plausible hypotheses to explain why we're seeing them. So here we see these plumes, right? And the other interesting thing is, if you were to take a look without that dynamical model to go with it at a static snapshot like this and see all these green cells kind of doing this together, you might hypothesize that this is kind of a collective behavior, like collective migration. But it turns out that you don't need collective migration to get these apparently coherent structures. That if you have every cell acting on its own, and doing independent motility, and just by stochasticity, one green cell finds a little crack, finds a little path of mechanical least resistance, it will push through and widen it out just a little bit to make it easier for the other cells to follow through. And so once one cell forges a path, the others will follow just because it's mechanically favorable to do so. So here's kind of what the simulation looks like. You kind of get the formation of this necrotic core and a kind of a ring that's been evacuated. And you see that these plumes grow into the surrounding normoxic tissue and eventually will invade the surrounding tissue. So you can try this model yourself online. We hope you do. And I also want to point out that we can take those assumptions we built originally into our 2D model and try them out in 3D just to make sure that it works. And we do indeed see the same behaviors in 3D. So that was a lot of fun. One last work I'd like to highlight is led by our postdoc, Michael Getz. And this is modeling SARS-CoV-2 or COVID-19. And so I'd like to really put a big thank you out. We actually are not doing this alone. It's impossible to do something like this alone. But we built a coalition of over 40 regular contributors across 20 institutions across the US and other places. Yafei also co-led this. And together, we got together and brought domain experts across immunology, virology, computational modeling, of course, informatics, and put ourselves together and combined this coalition of expertise to build an increasingly sophisticated model of COVID-19 landing in a tissue, the virus replicating in cells, escaping, and then the immune interactions. And so we built this up iteratively. First, we built an initial prototype that adapted some of our models from nanoparticles to add replication dynamics and build a basic model of viral replication. Then in our second version of the model, we added receptor trafficking so that the virus didn't just magically get into the cells. The virus had to bind to a receptor, get internalized, and was then rate limited perhaps by receptor trafficking. The third version of the model, which is really exciting, is when the community started building the rules of the individual immune cells, macrophage rules, neutrophil rules, T-cell rules. The fourth version of the model said, well, we know that this is not just a local phenomenon. We know that some immune cells will sample antigens, leave and go to the lymph node, help ramp up production of a T-cell army and start ramp up production of antibodies and bring them on back home to the tumor. Sorry, not the tumor, to the infected tissue. And so version 4 added this lymph node and this trafficking between the infected tissue and the distant lymph node. And then the version 5 model finally started adding the neutralizing antibodies. So some key interactions in this model is CD8 T cells that attack infected cells. And so when a T cell comes into contact with an infected cell, it will kind of form an adhesion, and then it will increase the damage in that attached cell by R delta T. So you have a damage rate times time, so that's how much damage you're creating. And then the infected cell has a basic Hill type like response model that says that you might have a base death rate, D naught, and then a maximum increased death rate, let's say 100-fold larger, and that the more damage you have, which comes from contact time, the faster rate of entering a program death. And then we might have macrophages as other rules that say when a macrophage comes into contact with a dead cell and realizes it has virus in it, it will phagocytose it, acquire the volume, and then start secreting pro-inflammatory signals saying, hey, I found an infection, we better call in the cavalry. And so let's kind of take a look at what this model does. So here you see kind of a field of blue. This is a bunch of initially uninfected epithelial tissue. And then the virus particles will land randomly in this tissue, start replicating in the cells, start releasing replicated virus that diffuses into the environment to infect other cells. And so you'll see the more infected the cell is, the more yellow it is. Macrophages will be scouring around looking for dead tissue, and when they find it, they'll start secreting pro-inflammatory factors that then start attracting, say, dendritic cells that will come and sample those infected cells, leave and go to the lymph node and report what they've seen. And then the lymph node will start creating helper T cells, or the CD4s, and the effector T cells, the red ones, the CD8s, that come back to the tissue and start attacking the infected cells to halt this infection. And there are other effects, of course, like interferons in here that we don't have time to go into. But let's kind of watch what happens here. So now we have some infected cells, activated macrophages attracting things in, see some infected cells replicating, but the immune system ramps up and brings the infection to a halt. So you can kind of see where the viral count in the tissue went up, then the infection was controlled, and now we have a lot of damage, probably some scarring in this tissue, but the infection has been brought under control. And so this model now has all the necessary pieces to ramp up that initial acute immune response, that innate immune response, go back and build up the adaptive response so that you eventually do bring the infection under control. And then here's the cool thing. I guess I don't have it in this one, but if you take the exact same tissue now with your pre-trained immune model, kind of wipe the slate clean, and so start off with another uninfected tissue, but now with the pre-trained immune cells and the antibodies, now sprinkle in it, like say, a secondary exposure, and this time you'll have only a very small amount of tissue death, and the tissue is rapidly brought under control. Kind of shows the notion that you can virtually immunize a tissue, either by a prior exposure or by exposure to antigens, and actually model that process in a multi-scale model like this. So we're really excited about that one. The other thing too, of course, is in modeling, you can take something you built in one problem and take it to another problem. So if you take the immune model we built initially for COVID-19 and replace the infected cells now with invading cancer cells, like these yellow ones here, you can have this notion of an innate immune and adaptive immune response responding to micrometastases, sometimes either reducing the growth rate of the metastases, sometimes even completely resolving them. So this kind of shows the notion of taking what you learned in one problem and transferring that knowledge to another one by taking your cell types and your cell hypotheses with you to another problem. So one last thing I want to point out before we wrap up here today is we do have an online resource available that you can use to explore as you're learning about PhysiCell and starting to play with your own models. It's called PC for Learning. It's an online interactive tool. So if you go to this link on NanoHub, you can register for free, get a free account. There's no cost to use this. And this will allow you to explore the key features of PhysiCell, to learn how to use the microenvironment to set up things like diffusion and decay, to set up your cell definitions, and then set up the basic phenotype, say the birth rate, the death rate, the size of the cell, the mechanical parameters. And then you can just click, run, and explore. It will sprinkle in some random cells and show you what it does. And so kind of to navigate to the app, you would kind of go to the page, click the Launch Tool button, and then you will be greeted by a user interface that has a number of tabs. We can learn about the overview of the model of this simulated environment. You can set up the size of the domain and config basics in how long you want to simulate. Microenvironments, where you start setting up your diffusible substrates like oxygen, glucose, growth factors. The cell types is where you're going to set your cell definitions and their default behaviors. And then you can click on Run, and there'll be an in-browser plot that you can actually look at the details. So I can show you just a really, really fast example. So here I've already entered into it. I'm going to just make, a small domain, let's say -250 by 250 in X. And I'm going to simulate not very long in time. Let's just say 6 hours or 360 minutes. And in my environment, I'm going to name my substrate quorum factor. So I'm going to use this from microbiology. Okay, so we have that. Let's maybe reduce the diffusion coefficient a little bit and a little bit of decay rate. I'm going to turn off my boundary conditions and just use zero flux conditions. So I'm going to start off with no quorum factor, low diffusion constant, low decay rate. Then my cell types, I'm going to rename this guy from default type to bacteria. And let's say we give them maybe a different cycle model, just a really basic one that we're going to say that I'm going to change it to duration. Let's say that they spend on average 180 minutes before they divide. So it could be a fairly fast proliferating cell type. And then for apoptosis, let's see here. Let's go like maybe 1/10 that. So 180, that's its proliferation rate. Divided by 10. So we're going to maybe do, it's not so far off, about 5e negative 5, 5e negative 4. Let's just go up. This will be our death rate. I'm going to leave the volume alone, even though it's unrealistic. I'm going to leave all this. I'm going to maybe turn adhesion off. Make this a non-adhesive species. I'm going to turn on motility, and I'm going to turn on chemotaxis, so they're going to move towards the quorum factor, so they'll start aggregating. And let's go ahead and just make them completely random in their motion. I'm going to make sure that they're actually secreting that quorum factor. So we'll turn that secretion rate to 10. I'm going to leave everything else alone and just click on Run. And in a second here, this is actually running in San Diego, so it takes a second before we see it. There we go. Now if I go to plot, I can start advancing through my frames. So you see I seeded some random bacteria here in my domain. It looks like I forgot to change one of my boundaries here. But you can actually click play to watch these cells that should be motile and aggregating together. And so you can keep advancing through the simulation this way. So this gives you a really fast test bed just to say, what happens if I change this or that parameter or turn on an interaction, and you start testing your model hypotheses online even before you get going. This might be a nice way to kind of learn and explore the capabilities of PhysiCell, and we'll show you a little bit of this as we go through the short course. So please do give that a look. Otherwise, what's new in the 2022 version of this course, we've tried to streamline the materials and kind of combine some of the lectures so you can get through it faster. We've added new built-in interactions into PhysiCell, particularly phagocytosis or predation, transformation, attack, and cell fusion, where they can actually fuse into one cell. We added these dictionaries I talked about, which we think is going to make modeling faster and easier than ever before. And we're starting to better integrate the intracellular modeling into the course. And so there will be a dedicated session on PhysiBOS, which is this beautiful Boolean signaling network that was developed by our colleagues at Institute Curie and at the Barcelona Supercomputing Center. And they will be teaching a section on how to build Boolean networks in your models. And then we'll also give a section on how to use LibRoadrunner to integrate ODE models into cells. And these are fully integrated. They can sample the environment, feed those into the intracellular models, run them, and then the output of the models will actually change the behavioral parameters of the cell phenotype. And we'll also have a few sessions on community-supported 3D visualization for the cells. So that would be really cool. You can access all of these materials on GitHub at this link or this humongous QR code. And so all the materials for this course will be posted here as we progress in the next week or so. I want to thank the Jane Koskinas Ted Giovannis Foundation for Health and Policy, in particular for a lot of funding for this work, as well as the National Institute of Health at the NIH and the Common Fund, and of course Leidos and DITRA for other recent funding. So thank you very much for joining along with us here today and for the next couple of weeks, and we really hope you enjoy this course and come to love modeling and PhysiCell as much as we do.